free airtime with #LearnOn1. Share your comments on the lessons. WhatsApp your feedback, name and town to 081 200 6659. #LearnOn1. Invite learning in.
Discovered the DNA molecule. Did you know that this molecule was discovered in 1969? That is not that long ago. Okay, perhaps a bit longer for you than for me. Now, we want to see that special code on the DNA that causes the code how to make a protein. And we call that specific part that is telling the body how to make a protein a gene. Yes, we are going to see how the genetics and heredity happens with the DNA molecule. So when we look at the history, it is important to know that genetics is the study of genes, and inheritance or heredity is how traits or characteristics are passed on from generation to generation. How do we inherit my mother's blue eyes? Or how can I make sure, if I'm a farmer, that the good traits or characteristics or quality of my show bull is passed on to the calves that he sired? Perhaps I am a vegetable farmer, or if I'm harvesting oranges each year, and I want the offspring to be just as tasty as the tree that I am giving water and pampering all year. How does that happen? Chromosomes are made of genes, and genes are built up of DNA. So when I am looking at the genetic material, I'm looking at genes and chromosomes and DNA, and we find that inside the nucleus of a cell. Gregor Mendel is considered as the father of genetics, and we are going to look at the reason why. So when I look into the nucleus of every cell, I will find the chromosomes. If you have to show me a picture of chromosomes, it will be a chromosome that has been replicated. So chromosomes sort of look like this. Now, it contains the genes, short parts of the DNA, and it is made up of DNA, thread-like parts that contains the genetic code for a specific protein is a gene. It's made of DNA. So, here I have 23 pairs of chromosomes. Pair number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23 chromosomes. 23 pairs of chromosome. This is called a karyotype. A karyotype is a photographic arrangement of a complete set of chromosomes in a human cell. So, we already did this, a bit of revision. A homologous chromosome means a chromosome from the father, a chromosome from the mother. And I would like to use symbols so that we do remember. Let's make the green one a chromosome that came from the dad, and the purple one that was part of the mother. When replication takes place, I have still a homologous pair of chromosomes. This is still coming from the father, but it replicated. And this came from the mother. Now I call this part a sister chromosome, and of course the centromere will keep the chromosomes, the two parts, the chromatids, together. So, when I am looking at a homologous chromosome, one of a matching pair of chromosomes, one inherited from each parent. So, this is a chromosome. And if I look at this picture, I have a homologous pair, but I can see this was not yet replicated. But that is not important now. What is important now is on this specific spot, which is called the locus, I find the color of the flowers gene. This is a gene for the color of the flower. On the homologous partner, I find the gene for the color of flowers. So this is telling me it's a plant chromosome. You agree? Now, on the one, it says purple flowers, and on the other one, it says white flowers. So we are talking of the alleles for flower color. The one allele states it is purple flowers. Other allele on the same locus will tell me what is this chromosome's gene for flower color. Please go and quickly revise what does the word allele mean so that you can define it. Right. So now when I have a homologous pair, and this homologous pair had replicated, I can see they are a pair, they are exactly the same size, and they will have, if this is the alleles for flower color, this will also be the alleles for flower color. It may not necessarily be the same. It can be purple and it can be white flowers. If we are looking at a human homologous pair and we say that this is the alleles for eye color, this will in the homologous pair be the one for eye color. They can both be brown eyes or they can both be blue eyes. Or this one maybe can be brown eyes and this one blue eyes. So, what is the color of this person's eyes? Is it brown? Is it blue? Let's see if we can discover something else. So, allows alternative versions of the same gene. The gene for a particular inherited characteristics resides at a specific locus or position on the homologous chromosome. So, if I look at this picture, this will be called alleles. Why? They are for the same trait on the same location on two different chromosomes. But if I have a spot there and a spot there, it will tell me that it is two different genes. It will code for two different characteristics or traits. Now, a genotype and a phenotype. We have to now see what is the difference between genotype and phenotype. Genotype is telling me what is on the genes. So, on the genes, I will have allow for green eyes. So, I think that can be eye color. But I have eye color here on the genes. That tells me the genotype of this organism. On this, I will have also a specific gene for body shape. And I will have a gene that is telling me that this organism has a specific number of legs. So, this is telling me on the genes what is this animal's traits. But the phenotype is what can I physically recognize about this organism. I can say it has green eyes. You agree? It has a blue body. It has 3 legs and it has wings. Okay, and this was all codes that was on its genes. So when I'm asking the genotype of an animal, I use codes. When I ask the physical phenotype, I say it has green eyes. But when I want the genotype, I will have to discover a bit more. So, if I take the gene for purple flowers, you remember we had a gene for purple flowers. Now we use a capital P to indicate the gene for purple flowers. Why capital? Because purple is the dominant trait. And then small p will mean what? It will mean white flowers. But I still use the small letter of the dominant trait. And white will then be the recessive gene. Okay, I really hope that you are still with me. So, I have here a dominant feature therefore I've written it in a capital letter. I have a recessive feature, therefore I've written it in a small letter. But I use the P because the dominant feature is purple. In this case, we say the recessive feature is white. And when I have a capital P, small p, gene combination, we say it is a heterozygous, different genes. This specific organism has two different genes for, let's use flower color. Okay, but what happens with these alleles? Locus for second gene containing alleles has a capital Y and a capital Y. Yes? So, we say it is homozygous in its genotype. Let's say that capital Y was for round seeds. Round seeds. And round seeds is dominant. Then it means the genotype on both the alleles are capital Y, so it has the same genes on both the loci, and therefore we say the genotype for seed shape is both dominant is round seeds. But it is heterozygous for flower color. It do have a purple dominant gene and a white recessive gene. Therefore, two different genes is a heterozygous genotype and a homozygous genotype. So, if we go back to our little explanation here, is the genotype will be a code, right? And the phenotype will be what we physically see. And now I wonder if this is the genes for this flower to have offspring. There cannot be only a male, but there's also a female. No, paternal, that is the dad's chromosome, and this is maternal, pa and ma, paternal and maternal, and these two are going to have babies. So, what will happen to the flower color of the babies if this is the mother and this is the father, and what will happen to the seeds? Okay, so definitions. Genotype, the types of genes or alleles that is present. What is the gene of the father? Purple. Dominant purple. What is the gene of the mother? White. Now, they want to have a baby, and these two genes will come together. Where? After fertilization took place, yes, the ovum and the sperm fuse and forms a diploid zygote. And now, the new offspring has a dominant purple gene and a recessive white gene. The question will always tells you which one is dominant, which one is recessive, or will give you enough information to deduce which one is dominant and which one is recessive. It's important to find that out. Now I know the genotype. of our previous slides offspring. But the phenotype is what will it physically, what will it physically look like when we look at that offspring. If a characteristic has same alleles, it is a homozygous gene or characteristic. If it is heterozygous, I know that it has a purple and a white gene. Right, heterozygous characteristic. Okay, so let's look at dominant and recessive again. Dominant means that this allele will be expressed if it is present. If a dominant allele is present in the genotype of the chromosomes, it will be expressed. And we always represent it with a capital letter. The trait we are looking at is freckles. Let's choose freckles for our letter to represent the dominant allele and the recessive allele. If you have freckles, it is a dominant trait. If no freckles is on the gene, it will be a small f. So recessive alleles will only be expressed in the absence of a dominant trait, and we represent it by a lowercase letter. Try to repeat. How do I define dominant? The allele will be expressed always when it is present, and I look for it or write it as the capital one. A recessive allele will only be expressed in the absence of the dominant one. Always expressed if it's present, only expressed if there is no dominant trait. Okay. So, a dominant allele is expressed even if it is paired with a recessive allele. Here, I have a phenotype. Phenotype. This cat is brown. Yes. This cat is brown. Yes. And this cat is gray. Okay. So, this is, now I'm looking at the genotype. This cat is homozygous, capital B, capital B. You agree? Its genotype is homozygous, brown. This one is homozygous, gray. Can this cat shows any other color than gray? No, because the recessive alleles is visible. Gray is recessive over brown. Okay, because I see small letters. If this cat is gray, it can only be homozygous for the recessive trait. But this one is, the phenotype is brown. But can I say it is capital B, or capital B, small b? I cannot say unless I have more information. Now, so this one is a heterozygous genotype. But physically, I will see only the dominant allele because it is always expressed, even if it is paired with the recessive allele. If I see the recessive traits, it means it is small b, small b. So, what did Gregor Mendel then do? He was an Austrian monk, and he lived in a monastery, and he started to experiment with pea plants, because he recognized that there are specific characteristics that occurs in some plants, and then in others it is gone. Why pea plants? They were available, they easily grew in the garden, they reproduce quickly, and they have differences in their traits. So, understood that there was something that carries traits from one generation to the next, and they call it a factor. During Gregor Mendel's experiments, he was not aware of the existence of genes and DNA and chromosomes. He only knows there was a factor that was available. There you see Gregor Mendel, 1800s. Okay. So, he started his experiments to apply the question on inheritance. For 7 years, Mendel bred pea plants and recorded inheritance patterns in the offspring. Particulate hypothesis of inheritance. Parents pass on to their offspring separate and distinct factors. Today we call it genes. Parents pass on to their offsprings separate and distinct factors that are responsible for inherited traits. Now, Mendel was very, very careful to make sure that there are no contamination. It is very difficult to know which plant is the father and which plant is the mother. Which flower had produced the pollen, because the pollen contains, not is, the pollen contains the male gamete. And inside the ovary of the flower is an ovule with the female gamete. So what he did was, he worked very carefully. He tried to enclose the plants that he was working with. He removed the stamens, in this case, from the purple flowers, so that this purple stamen will not produce pollen and self-fertilize this flower. He took that and he transferred it with equipment, delicate equipment, to this, the pollen from the white, and he put it on the purple ones. He knew exactly where did he do this pollination and made sure that the carpel matured, and then he planted that, because when you have this, you don't know how the offspring will look. No, this is only containing the embryo of the new offspring. So you have to plant it, take care of it, and now you have your first generation of offspring. So, purple and white. White was the father and purple was the mother. And the first generation offspring then was there. He used color of the flower, position of the flower, seed color, seed shape, pod color, stem length, all different features that he used. And it was very important to prevent self-fertilization. Okay, so he cross-fertilized his plants by hand. It was very important because he needed to control who was the parents in order to see if the parents transfer the genetic characteristics. Now we come to what will happen. The first thing is we are looking at monohybrid crossings. That means we are only using one characteristic at a time, not purple flowers and